Okay, I think we're going to go ahead and get started. Welcome, everyone. Really excited to see so many participants here, and I imagine we'll probably have even more join as the time moves forward. Thank you for joining us for the Children's Healthcare of Atlanta and Emory University COVID Force Collaborative Seminar. My name is Ann Sharudi. I'm the director of the Center for Childhood Infections and Vaccines, or CCIV. We are sponsoring this event along with the Center for Clinical and Translational Research. So just to give you a very brief background on COVID Force, we started in March of 2020. FORCE stands for Feasibility and Optimization of Research at Children's. And the idea and the mission of this COVID Force Collaborative was to provide strategic coordination to facilitate the best possible research and remove barriers to move COVID-related research forward. This seminar represents the fourth of this series that have started sort of more recently, since a lot of that research that started in March of 2020 has come to fruition. And the series is designed to share the important work of researchers within the collaborative and to those in our community. So just a couple of housekeeping items before we get started. We're going to ask everybody to stay muted. Please do not unmute yourself to interrupt and ask questions during the presentation. There should be plenty of time for questions at the end. I'd like to utilize the chat box for these questions, and I'll be moderating that throughout. So I'll try to, I envision we've already gotten some questions by email, and so I imagine that that will be a pretty lively session. So what I will do is moderate the chat throughout the seminar. Please just throw in your questions early. You don't have to wait until the end. And I'll try to group them into certain themes to allow Evan ample time to address each of the issues. So go ahead and put your questions in chat and I'll pose them to Evan at the end of his talk. So turning now to our introduction of our speaker today. I'm really excited to introduce a colleague and somebody that I have really enjoyed working with over the years. Dr. Evan Anderson is a professor of pediatrics and medicine here at the Emory University School of Medicine. He graduated kind of going back to the beginning, summa cum laude from Wheaton College, and then he pursued his medical degree at the University of Chicago Pritzker School of Medicine. He then stayed there for residency in both internal medicine and pediatrics. And he's also completed both an adult and a pediatric infectious diseases fellowship at Northwestern Memorial and Children's Memorial Hospitals in Chicago. So he's now board certified in internal medicine, adult infectious diseases, pediatrics, and pediatric infectious diseases, so a quadruple threat. He was really very well poised to address the COVID pandemic when it began back in 2020. Right now, he splits his clinical care time between adults and children, and initially started his career at Northwestern and then came to us, thankfully, at Emory and Children's in 2012. So here he's an attending physician at Children's Healthcare of Atlanta in pediatric infectious diseases and Emory University Hospital in adult infectious diseases. He is one of the multiple PIs of the Emory University Vaccine and Treatment Evaluation Unit, or VTEU, and he serves as the lead investigator in Georgia for influenza, RSV, and COVID-19 surveillance for the CDC-funded Emerging Infections Program. He's also, I can't go without saying that he's an excellent mentor for many residents, medical students, fellows, and has received the Emory Department of Pediatrics Research Mentor Award in 2017. He has over 150 total publications with an interest in rotavirus, RSV, influenza, COVID-19, early phase vaccine clinical trials, and the power of community protection. So with that, I didn't want to make that short because Evan really has done an amazing amount of things, and I just wanted to make sure that you all knew what a wonderful speaker we have today. But with that, I will turn it over to Evan for his talk, COVID-19 vaccines. How did we get here? Where are we, and where are we going?
All right. Hopefully you can see the presentation in slideshow mode.
It's in the presenter mode right now.
Let's see here. Does that fix the problem?
Perfect.
All right, great. Thanks, Sam. So thanks for the kind introduction. It's fun to look through those that are on the call and see actually a number of colleagues, friends, and actual participants in the clinical trials that I'll be talking about here. So it is kind of a three-part presentation, and hopefully you'll learn something in the midst of getting to hear about this. So here are my financial disclosures, including clinical research funding to Emory, served as a consultant for several different companies, served on the safety monitoring committee for several others, and then really these are NIH funded studies that I'll be talking about: Moderna, the phase 1 and phase 3 studies, and as well the Janssen Ad26 spike study. So first question is, how did we get here? So, so this is how we got here. Simply put, we as humans love our animals, and we are not particularly good at staying separated from them. And so we're sharing and swapping our viruses all the time. So the picture up in the upper left is a child that was made famous during the 2009 swine flu pandemic. Here on the right, this is actually a bat that this woman is kissing, and then of course a camel here. The reason that this is relevant is that animals have been a source of the coronaviruses. And so back in 2003, we saw the initial emergence of the first severe acute respiratory syndrome virus, or SARS, the original SARS virus. And then subsequently in 2013, we saw emergence of MERS coronavirus. And so we were somewhat prepped for the possibility of a new emerging coronavirus pandemic. Not as prepped as I think we would have liked to have been in hindsight, but in better shape than we might have otherwise been. One of the key things that was identified is that if we think about COVID-19 or SARS-CoV-2, one of the key proteins is the spike protein, which sticks off the inner ball of SARS-CoV-2. It was identified that that spike protein and development of an immune response against that could potentially provide some degree of protection against subsequent illness. So this was a study that was started in the pre-COVID-19, but then ultimately published post-COVID-19 by the group at NIH. And really one of the key things to emphasize here is that, we were in much better shape than we otherwise would have been had we not done as much basic science work as had been done previously. And so that really ended up paying off immensely in terms of being able to expedite vaccine development. Here you can see this was actually animals that were immunized with mRNA vaccine against MERS, and they're given various doses of the vaccine, and then they were challenged. And what you can see is that weight loss was dramatically greater in those that received placebo, here PBS, in comparison to those that received a moderate or a high dose of mRNA vaccine for MERS. As well, it also protected against having identifiable virus in the lungs, decreasing the amount of virus, and also preventing lung hemorrhage. And so this data provided a lot of the background framework for the FDA making a decision to actually go allow for the phase 1 and phase 2 clinical studies to move forward without having done SARS-CoV-2 specific studies. One of the concerns as the field began to develop was that of wanting to try and avoid enhanced disease associated with a vaccine. So this had been seen classically with the RSV vaccine in the 1960s, where there was vaccine-associated enhanced respiratory disease, where children actually became more sick instead of less sick after they'd received a vaccine. And this, of course, had really impacted RSV vaccine development for really 40 years. From that standpoint, that was one of the potential concerns based off of some animal data that was identified, particularly related to the feline infectious peritonitis virus and others within the coronaviruses. Fortunately, there's not been significant clinical data supporting this is actually occurring in humans to date. And we are seeing, importantly, a Th1 biased response instead of a Th2 biased response, which seems to have correlated with enhanced disease, particularly in that RSV study back in the 1960s. Here you can see the ball of SARS-CoV-2 with the spike sticking off. So this is spike protein. Within spike protein, it contains several, or one key area, which is the receptor binding domain. And that receptor binding domain is thought to be important in terms of its ability to fuse with the virus, to fuse with ACE2 receptors in particular. What you can see is that there's some homology between that protein and the original SARS coronavirus, and then very little homology with the other coronaviruses, the quote unquote endemic coronaviruses that are down here in the last 4 in particular. So that's in part why prior human coronavirus infection was not preventing against actual SARS-CoV-2 disease. The field of vaccine development for SARS-CoV-2 is a good bit dizzying. So there are over 40 different vaccines in clinical trials currently, with over 150 in preclinical evaluation. So how do you make sense of that has been a key question. And really what I'm going to focus the vast majority of the rest of my talk about is the efforts that were put forward by the US government to develop one or multiple vaccines for COVID-19. And in essence, this was an investment by the federal government where they took vaccines from using several different strategies from several different manufacturers, and essentially funded them to pursue those vaccines at risk, and said, we will buy 100 million doses of vaccine from you in January. You need to produce it by roughly about that time, but we'll plan on purchasing it regardless of whether or not you actually are successful in moving your vaccine all the way through to successful licensure. From that standpoint, I think this investment actually ended up being about $26 per person here in the US. So it was not a huge dollar amount, actually, if you think about it on a per capita basis. And what they did was they funded 2 mRNA vaccines, the Pfizer and Moderna vaccines, 2 viral vectored vaccines, the AstraZeneca and the Janssen vaccine. So these are using other viruses. Here for AstraZeneca, it's a chimp adenovirus vaccine. For Janssen, it's an adenovirus 26 vaccine. And then there are 2 protein-based vaccines, Novavax, which just started their clinical trials in December, and the Sanofi, which was delayed. And there's been some public word that there's some they had insufficient antigen present in their phase 2 study, so they had to go back and repeat some of the early work. And what you should be aware of or get the sense for already is that these protein-based vaccines are moving much slower than some of these other newer methods of vaccine development. Of course, protein-based vaccines being some of the classic strategies for vaccine development. So one of the sustaining or encouraging things in the midst of the COVID pandemic has been humor. And this is from The Onion, which is very tongue-in-cheek. Their headline was, "Nation can't believe they spent so much time overlooking the obvious solution of mRNA instruction for spike protein encapsulated in a lipid nanoparticle." So that of course is meant to be humorous. This is not something that's intuitive to most people, and so it should not be surprising to you that you're unfamiliar with this approach. So here I have things, here are things pictorially presented. So in essence, what they do is they take RNA, mRNA, which codes for the spike protein of SARS-CoV-2. There are 2 minor mutations that were made in that to stabilize the protein itself. And then they encapsulate that within a lipid nanoparticle, which in layman's terms is literally a fat globule. Is what that is. It's then injected into the arm, where it's taken up either by muscle cells or antigen presenting cells. It is then essentially the mRNA within the vaccine is then released into the cytoplasm of the cell. And for those of you with less science background, our bodies are continuously making mRNA all the time. It's what essentially drives protein manufacturing by our cells on a regular basis. There's also mechanisms to degrade that mRNA that are very, very active. So what happens is that mRNA is released and immediately the cell can't tell it's any different than mRNA that it would see that it itself is making, and it begins to make protein. And then it is presented to the immune system either through MHC class 1 or MHC class 2 presentation to stimulate CD8 or CD4 cells, generating hopefully an immune response. Again, this is in part to provide you some overview. If we think about some of our classic vaccines, such as live attenuated vaccines with LAIV, rotavirus and MMR varicella, this strategy has not moved forward very quickly here in the US, in part because quickly attenuating a virus is challenging. Inactivated vaccines have also not moved forward quickly. That would be viruses like hepatitis A or the inactivated polio vaccine. Again, due to issues of concern, in particular related to the inactivated vaccines and potential disease enhancement. And so instead, these other strategies have been the primary ones to move forward. Here's pictorially where the current leading vaccines are among those 6. Of course, Moderna and Pfizer are kind of sitting in between, still finishing up their phase 3 trial and yet have ongoing phase 4 data that they're starting to begin to look at. AstraZeneca and Janssen and Novavax are all in their phase 3, and Sanofi Pasteur has kind of moved back to phase 2. When we think about developing a vaccine, we need several things. So first, we do need immunogenicity, meaning an immune response from after the body is exposed to this antigen. We also need to quickly develop a vaccine but maintain safety. Safety is critical as vaccines are given to otherwise healthy people. Especially in the phase 1 studies, once you administer, well, essentially anytime you administer a vaccine, you can't take it back. And so particularly for the phase 1 studies, those move very slowly as we enroll small numbers of individuals, frequently pause enrollment, and slowly dose escalate the vaccine. A key thing to highlight that many people don't think about that aren't thinking actively about vaccines is that safety is not necessarily the same as reactogenicity. When we say safety, we often talk about lab or organ toxicity, severe events, hospitalizations, whereas reactogenicity are things that we expect to see. That's local and systemic side effects from the vaccine. We expect to see reactogenicity as the body starts to develop an immune response. So you've heard people say, oh, I got the flu after the flu vaccine. Well, that's the immune system turning on and beginning to generate its immune response to the vaccine. Sometimes reactogenicity and safety can overlap, such as having febrile seizures or with the whole cell pertussis with the high fevers and neurological complications. In the recent past, a general philosophical approach has been to minimize the reactogenicity of our vaccines. However, and I think that this kind of paradigm has to be rethought to some extent when we have a potentially life-threatening disease or a severe disease that we really want to try and prevent. Safe vaccines can have substantial reactogenicity. Safe vaccines actually can have substantial reactogenicity and yet can have a better immune response. And the classic example for this is Shingrix. For those of you that are older, you may have received Shingrix, and you know that that vaccine can absolutely pack a wall up as far as local symptoms of pain, redness, lung tenderness, soreness at the site of injection, and then also the systemic side effects of fevers, chills, achiness. A lot of people end up having to take a day or days off of work after receipt of vaccine. So if we think about the two extremes, with flu vaccine not being particularly effective, maybe 50% on an average to good year, and Shingrix, which is more in the 95% efficacy range, but much more reactogenicity, the vaccines that we'll be talking about in general are a little bit more shifted towards that of Shingrix than that of seasonal flu. So we conducted a phase 1 study, and we will end up focusing a little bit more on the Moderna vaccine, in part just because of our personal experience here, of a dose-ranging study of the safety and immunogenicity of this mRNA vaccine in healthy adults. It was initially planned to be 3 arms, 15 individuals in each. But what had happened, if you remember, Kaiser Permanente had been selected to be the lead site. COVID-19 emerged in Seattle at the time that they were getting ready to start the study. And so in essence, we were asked to provide backup to Seattle on March 12th, which for those of you that were here at the time, it's essentially the day that Atlanta ground to a halt and many kids went home from school to stay home from school for the next long while. There was a real need to move at warp speed. We essentially were able to get biosafety approval same day, submitted to a single IRB, and were site activated on the 21st. We began screening on the 23rd. Importantly, because of all the stay-at-home orders, people essentially stopped flying. And so the actual transportation infrastructure changed in between the time that the study started and the 27th of March. So it was actually a couple-day delay in terms of actually flying the vaccine down from the DC area to Georgia, because there were no flights going. And so it ended up having to be driven down. We ended up enrolling into additional cohorts now over the course of time. Additional challenges for us were who was previously infected or infected at baseline. Again, if you remember back to that time, the PCR testing was just becoming available at a state level for patients that were hospitalized, but was not routinely available at all. Antibody tests had also not been developed at the time, and certainly not commercially licensed. So in essence, we had to move forward based off of history alone without actual testing at baseline in our participants. There's ongoing risk of COVID-19 infections during the trial that could impact both the safety and reactogenicity of the vaccine and also the immunogenicity of the vaccine. And so trying to do everything we could to minimize the risk to participants and also the staff that were in a close setting with these individuals for 2 to 3 hours. Importantly, if you remember back to that time, there was no universal mask policy. It wasn't clear to the extent that, that things were transmitted through the air and through droplets. And we actually had insufficient masks with no certainty of resupply. But we made a decision to move forward with having our staff and participants masked, hoping that we might get a resupply before the third or fourth subsequent study visit. And for a while, we're actually primarily dependent upon homemade masks that had been donated. There's no other available PPE. There were work and travel restrictions that, that were threatening to be enforced that could result in staff having trouble getting here and participants having trouble getting here. And then, of course, everything had changed in terms of childcare and schools and so forth. Additional challenges included just incredibly inflexible timelines. We had to meet deadlines in order to keep the vaccine on movement forward. For our phase 1 studies, it's important to note that these were vaccine only. There was no placebo arm. So as such, anyone attributed anything to be associated with the vaccine in general. And there was a tremendous amount of media craziness where the public eye was on literally everything that was going on. Participants were going directly to media with describing their reactogenicity and so forth. And then, of course, what has now become commonplace is publication by press release, which, of course, many of us are not enthusiastic about, but such has become life in the time of COVID-19. This was one participant in Seattle in particular who documented very clearly in his reactogenicity after getting the 250 dose of vaccine. So we looked at 3 different doses of the vaccine. And in essence, what you can see here, so for any systemic symptom with increasing dose, you see in general increasing systemic symptoms and more severe symptoms. Also, local symptoms were more so with the second dose than the first dose and increased with increasing dose. And so really based off of our experience in the trial and the participant I just alluded to, a decision was made to drop down from their planned 250 dose to 100 microgram dose, which is great on a number of levels. In essence, we now have 2.5 times the number of doses available immediately by that change that was made, as we realized that the 250 dose was not sufficiently well tolerated for individuals that received it. Move on to the next slide. So one of the challenges with understanding the immune response to the vaccine is, what do you measure it against when there's no accepted standards and when these assays are just literally being developed at the same time that the studies are being conducted? And so you can see here in our initial publication, S2P, so this is the spike 2P immune response, and then receptor binding domain. These are just binding antibodies. In essence, you see response immediately after, by 2 weeks after the first dose of vaccine, that then boosts up with the second dose of vaccine at day 36. So that's a week after the second dose of the vaccine. Notably, these are on a log scale for antibody titer. So that's why they look like there's not a huge difference, but it's actually a substantial difference, about a 10-fold difference in terms of the antibody. Neutralizing antibodies, which of course are the kind of the gold standard, and PRNT assays are very time intensive and very challenging to do. Hence, there's not as many kind of data points, but you can see that neutralizing antibodies were developed. And then as well, pseudo-neutralizing antibodies were developed in recipients. And really, one of the other key things to note is that there wasn't a huge difference between the 100 microgram dose and the 250 microgram dose as far as the immune response. And it compared in general fairly favorably to the convalescent plasma, which is the kind of the best measure that most manufacturers have had up to this time point as far as immune response. We then moved into the 56 and above age groups here, 56 to 70 and 71 plus. You can see in general that, that similar to the younger adults, you see increasing reactogenicity with the second dose, and there is some severe systemic symptoms after that second dose. And then as well, local symptoms are generally worse with the second dose than with the first dose, primarily pain at the site of injection. Vaccine was administered on days 1 and 29. And here in these older adults, again, here are binding antibodies and pseudo-neutralizing antibodies. In essence, you need that second dose to really achieve good pseudo-neutralizing antibodies. And this is also evidenced by the FRNT assay that Mehul Suthar here at Emory has developed and is now being pretty extensively used in evaluating the, looking for correlates of protection as a much faster neutralizing assay than the classic assays that like PRNT. Importantly, the T cell response that was observed is really a Th1 biased response here. So this is Th1 cells, response in Th1 cells. You can see that you see in general a better response in terms of any Th1-like response with interferon, IL-2, or TNF-alpha, and relatively low Th2 responses, which is the type of immune response we were really looking for. So here's then data from the phase 3 study. Notably here we have a placebo arm. And I think that, you know, placebos are really quite helpful in a lot of different ways. You'll see that, of course, about 20% of people actually had symptoms after receiving normal saline, helps to balance out the side effects that we're seeing in the vaccine arms. But clearly there's more reactogenicity in those that receive the vaccine, including some severe local events, particularly that being of pain at the site of injection. For the systemic side effects, which are presented here, these are primarily headache, fatigue, myalgias, and arthralgias. Again, in the placebo arm, you actually are seeing some individuals with up to about 40% of individuals with some symptoms after receiving placebo, including some severe events after receiving placebo. And then clearly much higher reactogenicity after the second dose than with the first dose. So what happens then in these phase 3 clinical trials, and I just show here a picture of me in the midst of obtaining blood from a subject, is in essence, when they had COVID-like illness, they then were brought in and had nasopharyngeal swabs obtained and blood obtained. And in essence, we were blinded at the time, and you follow for the number of cases and those that received the placebo versus those that receive the vaccine. And you can see that right about day 14 or so, there begins to be a clear divergence of the two graphs. And that divergence continues to increase over the course of time. In essence, after that first dose, you're looking at probably about 50% efficacy. And by a week after the second dose, you achieve the 94, 95% efficacy that was observed. Then we'll shift gears over to the Pfizer vaccine. For Pfizer vaccine in general, the responses were fairly similar as far as reactogenicity to that seen with the Moderna vaccine. So this is their phase 3 clinical trial, and they have it split by younger and older adults. The good news is if you're older, in general, your reactogenicity is probably a little bit less, but again, primarily pain at the injection site. Systemic symptoms tend to be fatigue, headache, chills, and muscle pain. Again, worse with the second dose than with the first dose. And then when you compare those that are younger, 16 to 55, versus those that are older, in general, less reactogenicity as we get older. Here they have an inset figure here such that you can see that kind of separation really occur a little bit better. So again, numbers remain fairly similar until about the day 14 mark, where you then see some separation beginning to occur in those that receive placebo versus those that receive vaccine. Again, 50% efficacy after the first dose, primarily after that day 14, and then to achieve the 95% efficacy, you need a week after the second dose. This highlights some of the differences between the vaccines. A couple of things to highlight here, of course, as people have heard, and it continues to be talked about, the freezer issue for the Pfizer vaccine. Timing of the vaccine doses is different. And then importantly, you can see late redness and swelling occurring a week or even later after receipt of the vaccine. This has been seen in multiple individuals, less than 1%, but definitely seen frequently. You can see some regional lymphadenopathy with this as well. One of the safety signals that's being followed on an ongoing basis is that of Bell's palsy. So there were 4 cases observed in those that received the Pfizer vaccine versus 0 in the placebo group. For Moderna, it was 3 and 1. Right now, there's not thought to be a clear association at the present moment, although that's being monitored on an ongoing basis. And then, of course, allergic reaction in those with pre-existing conditions. Importantly, some individuals, because of the reactogenicity, will have symptoms similar to COVID-19 after dose 2 in particular. And then again, just mentioning that the reactogenicity is a little more similar to that of Shingrix than that of seasonal flu vaccine. So where are we now? So I'm going to take a few minutes and just highlight where we're at right now, and then as well where we're probably headed over the course of time here. So EUA was provided for Pfizer vaccine. Notably, they're 16 and above with their EUA. Those that dissented from the decision were primarily dissenting because of the issue of the 16 and 17-year-olds that were in the study. A number of them felt like there weren't enough 16 and 17-year-olds to license it down to that group. It's a 2-dose series, 3 weeks apart. You should not receive it if you've had allergic reaction after prior dose, or if you have allergic reaction to either of the components here. Notably, polyethylene glycol or sorbitol seems to be associated with allergy to that, seems to be associated with these allergic responses that are seen occasionally. Moderna has also had a few cases of allergic response also identified. Again, PEG seems to be the issue in particular. I'll highlight the fact that the ACIP, or Advisory Committee on Immunization Practices, routinely puts out recommendations of how to utilize vaccines after licensure. And these should be considered guidelines and are actually quite helpful and answer a lot of the key questions. And I apologize, the next few pages will be fairly text intensive. There's not an easier way to really do it than just power through it. In essence, you shouldn't receive vaccine earlier than 4 days before the recommended date, but you can, if they are administered earlier, they don't need to be repeated. There is no maximum interval between first and second dose, although typically we would prefer for them to be administered roughly 3 weeks for Pfizer and a month for Moderna. The safety and efficacy of a mixed schedule has not been evaluated. We recommend avoiding administration of other vaccines in the 2-week window around each dose of vaccine. They are safe in people with prior evidence of a COVID infection. So this is important. To join the study, you did not have to have a negative antibody previously. to SARS-CoV-2. And so when they looked at the safety in those that had had prior COVID infection, the safety was similar to those that had not had prior COVID infection. If someone has acute COVID infection, they do need to have their dose deferred until they've recovered. And then the recommendation here in the text here is fairly clear, but creates a lot of confusion in general. The current evidence suggests that reinfection is uncommon within the 90 days after the initial infection. Thus, persons with documented acute SARS-CoV-2 infection in the preceding 90 days may delay vaccination until near the end of the period. But they are, in essence, recommending vaccinating within that 90-day period. But if you want, you can delay out to 90 days. That's the recommendation. Okay. Then the other point to highlight there is that if you had received a monoclonal antibody or convalescent plasma, you should wait till 90 or more days. For those with a prior history of Guillain-Barré syndrome, there is there's no precaution or contraindication to receive a vaccine. For those with a history of Bell's palsy, because that had been noted in the clinical trials, although not statistically significant, but they're monitoring that on an ongoing basis, it's not concluded whether there's any causality. For pregnant women, and this is an issue, so a number of us were advocating even a while ago now for pregnant women and for pediatric studies to begin. That has not happened in essence to any appreciable extent yet. And so we're seeing a delay as far as having data for our pregnant women. So the way that they phrase things is that importantly, if COVID-19 can be associated with severe illness in those that are pregnant, and that risk appears to be even higher in the third trimester, potentially, including preterm birth and even fetal mortality. So it is based on current knowledge, mRNA vaccines are not thought to pose a risk to the pregnant person or to the fetus. These are not live vaccines. They're also degraded quickly by normal cellular processes. If a pregnant woman is part of a group that's recommended to receive COVID-19, they may choose to be vaccinated, but it's a conversation between them and their healthcare provider. Okay, and that should encompass kind of the risk of transmission, individual risk of contracting COVID-19, and so forth. We do hope to be performing actual clinical trials of COVID-19 vaccines in the near future, but those have not started yet. For lactating women, there's no data, but these are not thought to be of any real risk for a breastfeeding infant because they're locally administered. The mRNA is taken up locally and the protein's produced locally. So there should not be any real risk to breastfeeding infants. For immunocompromising conditions, those with stable HIV were included within clinical trials. Data is limited. Immunocompromised individuals may receive COVID-19 vaccination if they have no contraindications to vaccination. However, they should be counseled about the unknown vaccine safety profile and effectiveness in those that are immunocompromised. And they'll need to continue to protect themselves against COVID-19. Antibody testing is not recommended to assess for immunity after COVID-19 vaccination, and there's no current recommendation to revaccinate should immunocompetence be regained. These recommendations may change over the course of time as additional information becomes available. Autoimmune conditions, those who are autoimmune were eligible to enroll in clinical trials. There are no imbalances observed in the occurrence of symptoms consistent with autoimmune conditions or inflammatory disorders in those that receive vaccines. So I'll briefly hit on the other potential vaccines that are continuing to move forward here in the US. There's AstraZeneca, Janssen, and then the adjuvanted protein vaccines of Novavax and Sanofi Pasteur. So the chimp adenovirus or AstraZeneca vaccine had been one of the front leading candidates, but had run into an issue in which a participant had developed a transverse myelitis. That issue has been resolved, but their enrollment had been markedly delayed. They also, so they published now in Lancet in December some data about the efficacy of the vaccine as a whole, where they saw 70% efficacy. However, it's a bit confusing because essentially there had been a group of individuals who had received a lower dose of the vaccine followed by a standard dose of vaccine by accident. That in that group had much higher efficacy than the remaining cohort. So this remains to be sorted out exactly what was going on here in these clinical trials overseas. There is a phase 3 study here in the US that will provide US data. And I think the FDA is going to take a close look, not only at their US data, but also their international data to try and make sense about what, how and if they should license it. For the phase 1 adenovirus 26 spike protein COVID-19 vaccine. So this was literally just published on Wednesday this week. A few things to highlight. In essence, this is low dose, high dose, and placebo. The low dose is what has moved forward in the US. You can see that systemic symptoms and also local symptoms were less in those that had the lower dose, which is the one that's moving forward, than the high dose. Again, fatigue being most frequent, fatigue, headache, and myalgia being the most frequent symptoms. Pain as well, the most frequent local symptom. Their immunogenicity data is a little busy, so to speak. I put a line over the dose that's moving forward in their phase 3 clinical trial, so single dose of low dose vaccine. And you can see here, these are their antibodies that are achieved. There wasn't a huge difference between that and the high dose vaccine, and they decided to go forward with just a single dose of vaccine. Their convalescent plasma is over here on the right. And importantly, the convalescent plasma in one study cannot and should not be compared with the convalescent plasma in other studies. It will remain to be seen whether one dose results in similar immunogenicity with the Janssen vaccine. The protein-based vaccines, so the Novavax is now also moved forward into phase 3 clinical trials. It's their middle bar here, and you can see in essence they had, with the second dose of vaccine, more reactogenicity than with the first dose, primarily pain and tenderness at the site of injection. For the Novavax vaccine as well, more systemic symptoms with the second dose than with the first dose, primarily fatigue, headache, myalgia, and malaise. Their immunogenicity responses are here. These are using different assays than the ones that are used in the other published trials. Essentially, you can't compare across trials is one of the key things that I should emphasize. But in comparison to their human convalescent plasma, they saw good immune responses, both in terms of their binding antibodies and then also their microneutralizing antibodies. Sanofi had a major setback in terms of their efforts to develop a COVID-19 vaccine, and it's publicly been stated that it was due to the antigen concentrate amount of antigen in their vaccine was resulted in their need to move backwards rather than forward. So looks like this is going to be delayed a bit. So where are we going? So there's some key residual questions from the phase 3 study that we do hope to have more information about and really are looking forward to over the next days and weeks and months. So longer term safety. So currently we're at a median of 4 months for the Pfizer vaccine and for the Moderna vaccine. For the phase 1 study, however, we're now out at 10 months for some of the participants in that study. The durability of the immune response remains to be definitively shown. We were able to demonstrate in the phase 1 participants that, that there is a decline in the antibody titer, but it's not a huge decline, and it's still at or above that of convalescent plasma 3 months out from receipt of vaccine. There's questions about need for a boost and then the safety of a boost dose of vaccine, meaning a third dose of vaccine. Will the vaccines be able to blunt viral replication and transmission in vaccine failures? So this is a key question. So non-human primate data look really very, very encouraging. So this is data similar to what I had shown you at the very beginning, but this is with SARS-CoV-2 in non-human primates where they vaccinated them and then challenged them. And in essence, in those that received a challenge, you can see in comparison to placebo here, there's much less RNA in BAL fluid and also in the nasal swabs in those that receive vaccine versus those that received the PBS placebo. So that suggests that that may well occur. This was also demonstrated for the Janssen vaccine. I'm not aware of any data for the other vaccines, but that's really encouraging data from animals. And then what was seen in those in the Moderna study, pre-dose number 2, everybody had NP swabs obtained. And in those that had received vaccine versus those that had received placebo, and in essence, there were more individuals who had were carrying COVID after a single dose of placebo in comparison to those that had received vaccine. And so that does suggest that we will be blunting transmission of the virus with the vaccines themselves. We do hope that there'll be additional efficacy data as well coming out, particularly looking at the correlates of protection. So as I alluded to previously, I think there's a big ongoing question. Will we even be able to conduct phase 3 clinical studies in women, or will we end up having to look using a different approach than your classic randomized controlled trial in pregnant women? For immunocompromised patients, what's the timing and the dose? Do you need a boost dose of vaccine? Those are questions which are not currently answered. For the variants, there's always the potential for viruses to escape from immunologic control. And so classic example of where we see that is influenza, where it continues to migrate away from our vaccines over the course of time. So you may remember a few months ago, there was a lot of concern about the D614G mutation, which had become predominant in the population. And here, in essence, from our phase 1 clinical trial, here's the original strain, and here's actually the mutated strain. You actually get better immunogenicity against the mutated strain than you do against the original emerging strain of SARS-CoV-2. So just because you have a mutation, that doesn't mean you necessarily lose immunologic control of the virus. Pfizer has some early data that they said looked like there was no evidence of loss of efficacy of their vaccine. We're of course waiting more full and complete data. We would like to see assays and sera standardized and ultimately correlates of protection identified. And hopefully these will be identified from those phase 3 clinical trials. This would really allow for better comparability across the vaccines and also the potential to license other vaccines without having actual efficacy data. It's unclear at the moment what other vaccines will be sufficiently successful to be licensed. Moderna and Pfizer really set a high bar. So originally FDA was wanting manufacturers to be at 50% or better. They're at 95%. So if a vaccine manufacturer comes in with 60% efficacy or even 70% efficacy, will FDA still say, you're good to go and you will license this vaccine, or will they change the bar, so to speak? And there is a need for vaccines to meet the worldwide need. We need cheaper and more stable vaccines that can be distributed worldwide. We also need to consider other approaches such as inactivated vaccines. And there's ongoing trials, as I mentioned, over 40 different vaccine candidates are in clinical trials. I'd like to finish up then with, do we need a vaccine in children? So if you remember back historically, what you were hearing back at the beginning is that children don't get sick and die from COVID-19. If you look at the data for that, a lot of that actually came from this initial publication from China in JAMA, where only 2% of their initial cases were in children. However, when we turn to the US, we actually know now that there's a substantial burden of hospitalizations in children. So that's been documented multiple times. There does seem to be a higher rate of hospitalization in those that are Black or Hispanic. In addition, we've identified the multisystem inflammatory syndrome in children that is a life-threatening complication after COVID infection. Here are listed then the current hospitalization rate in children from COVID-19. We're now at 34 per 100,000 in those 0 to 4 years of age and 20 per 100,000 in those age 5 to 17. This is now at the rate of kind of the lower limit of the past 4 years for influenza and also in both of the age groups for influenza. It's also much greater than the hospitalization rate for hepatitis A, rubella, for which there was very few hospitalizations, and varicella, we're now approaching to beginning to exceed the rate of disease resulting in hospitalizations from varicella. So when we think about these other viruses for which we now have safe and effective and routinely recommended vaccines, it sure would make sense to develop a vaccine for COVID-19. As well, mortality burden. And this is, again, from the pre-vaccine era. Roughly 50 kids a year died from varicella, about 17 per year from rubella, 3 from Hep A, rotavirus 20 to 60. Flu ranges from 110 to 192 within the past 4 years. We're now at 182 in the roughly 10 months since SARS-CoV-2 emerged here in the US. So we're seeing actual mortality associated with COVID-19. As well, this huge non-medical direct impact upon children of COVID-19 in terms of education, extracurricular activities, economic, emotional, psychological development. There is no doubt that there is absolutely a need to develop a vaccine for children to get them back to living life. So why do we need pediatric studies? Well, there are differences in height, weight by surface area, muscle mass, fat distribution. So really the dose may well be different in children than it is in adults. Initially, there had been a single expansion of the phase 3 enrollment in the Pfizer vaccine down to age 12 now. Moderna has also started a 12 to 18-year-old study at their commercial sites through PPD. Unfortunately, the response has been, in general, we'll begin pediatric studies after the safety and efficacy is established in adults. In the meanwhile, unfortunately, we've seen a lot of morbidity and even mortality in children. We know from multiple other vaccines, actually, phase 2 and phase 3 studies in children usually occur without large studies of adult safety and efficacy. And if we think back to our history of polio, PCV7, PCV13, Hib, rotavirus, none of these had adult safety and efficacy established in large numbers of individuals before we began the pediatric studies. But the bar has been different for COVID-19. There are 2 studies here in the US, one with the Moderna vaccine and then the Pfizer vaccine that are kind of 12 and above. And just yesterday, actually, they had stated that they're struggling to get enrollment to their COVID-19 studies with Moderna. So the search for a vaccine does continue on. This is a picture taken from the phase 1 study of one of our participants and myself. And one of the things I'd really like to just express is a tremendous amount of gratitude for our participants that have been involved with these phase 1 through phase 3 studies. Just tremendous support from them in particular. Incredibly dedicated group of people that have been willing to take risk associated with receiving a vaccine in order to advance vaccines in general for all of us. And so very, very grateful to them for that. And I think I will leave it there. Any questions?
Thank you so much, Evan. That was really a whirlwind tour, but super, super valuable. There have been a lot of questions that have been coming in, and you have addressed actually a decent number of them. So I will try to, there were a couple at the beginning that are sort of related, so I'm going to merge a few of them together. So there were some questions, and you sort of touched upon this a little bit, but maybe you can go into a little bit more detail about kind of the 1 dose versus 2 doses and how thinking about vaccine effectiveness or efficacy, as well as reactogenicity, and whether, you know, what you're seeing after 1 dose, how that compares to what you see after 2 doses. And then as a kind of a adjunctive question to that, whether there's any differences in reactogenicity or VE, if there's any data on people who've actually had COVID before and then get the vaccine.
Yeah, so we don't really know vaccine effectiveness in those that have had prior COVID. We do know that in general, especially for more mild cases of COVID, meaning not hospitalized, that we see a substantial decay in terms of the immune response after initial infection. And so that's in part why vaccination is recommended. I think maybe I'll try and address this in general. I think I know where it's coming from. So there are discussions right now, and you probably heard it in the news, that in essence, all doses of mRNA vaccine would be released. And Biden's administration, incoming administration, had really wanted to change the approach here to try and get out vaccine much more quickly to everybody, with the potential that there could be delay between people receiving their first and their second dose of vaccine, longer than the window that was studied in the phase 3 clinical trial. So there's kind of an ivory tower answer. The ivory tower answer, as I sit here, is they're studied at 3 and 4 weeks for vaccine administration. So that's where we know they're most effective, and that's where we know we achieve that 94, 95% efficacy a week after the second dose and following. That's kind of an ivory tower strict interpretation of the clinical trials. But I think if you put it into the context of a nation that's really struggling to deal with COVID-19, it's a lot more complicated. And in essence, we know that a single dose, probably you're around 50% efficacy of the vaccine. And that's, that's actually quite remarkable. We don't know the extent to which that may also attenuate the severity of disease, even a single dose. So that may well be of additional benefit. And then, as I mentioned back before, there's some real evidence from those that were in the Moderna study at the time of their second nasal swab at the time of their boost dose of vaccine, that far fewer of them were actually carrying asymptomatically COVID-19 that had received vaccine in comparison to those that had received placebo. So if a single dose of vaccine can blunt transmission, then this may well be a more effective population-based strategy than trying to get everybody 2 doses as quickly as possible, but get everybody a first dose and then second dose as soon as that can be done. So it's a little bit of a philosophical difference. We do know that in general that actually the immune system tends to be more robust in its immune response with waiting a longer time period between the first and second dose. So it's almost as though it has additional time to reset and then develop a more robust immune response. So there may actually be some real benefit to waiting, but we just don't know specifically for the mRNA vaccines.
Okay, thank you. Sort of one other mishmash of questions put together is, and you alluded to this a little bit, and this is more probably speculation at this point, but can you talk a little bit about what you think the duration of immunity may be for the various vaccine constructs, or how that might differ between the various vaccine constructs, and how that compares to what we think may happen from natural immunity following COVID-19? And then as a corollary, maybe you can throw in a little bit of a comment on the UK variant and other variants that are emerging, and how these vaccines may address them, and whether we'll need new trials related to new products that are addressing those variants or not.
Yeah, so maybe I'll take the variant question first. So, just developing a mutation or even a couple mutations in and of themselves is probably not going to be enough to result in escape of the virus from a vaccine. Again, here, these were data that were for the initial really worry of the D614G. Pfizer has generated some data about that they released, although it sounds like it was fairly small numbers, that they're still seeing a good immune response against these variants that had emerged. So we don't know for the Moderna vaccine. We don't know for other vaccines about whether that will also hold to be true. There are very active investigations to try and answer that as quickly as possible. I don't think that there's going to be in essence, these vaccines are stimulating broadly the immune response to COVID-19. It does seem to be not just a single antibody clone, but actually multiple antibodies that are providing the binding and the neutralizing effects that are observed. And as such, I don't think that they're going to see a lot of escape from the vaccines. One of the upsides is that the improvement in the technology here is that with mRNA vaccines, you literally could change the vaccine overnight by changing the sequence that you put in with the mRNA to address a escape mutant, which is incredibly flexible. Now, the question would be, would FDA require a repeat phase 3 clinical trial of a vaccine that had a change of a single amino acid or a couple amino acids? They may well require another phase 2 or phase 3 study, fairly easy to conduct that relatively quickly, but there's not really great precedent except for flu, which is a good bit different as to whether or not they would accept a minor change like that without doing a big safety study. The durability in general, so we know we had published our data through 3 months after the first dose of, after the first boost with the Moderna vaccine. We don't have longer immunogenicity data with the Pfizer vaccine. And then just earlier this week, the Moderna president actually said that they expect it'll be at least a year of, or a year or longer as far as the immune response. So that's, I guess, public domain. I think it will be, yeah, at least a year or longer. Good chance it may well be longer. Although, you know, we'll hopefully generate some of that data from the phase 1 study in particular as we begin to understand the correlates of protection as well.
All right, thank you so much, Evan. I think we're past the hour, so we're going to end it here. I really appreciate it. I know everybody on this Zoom appreciates that the time that you took away from all the important work that you're doing to share your expertise and insights. Thank you to everybody for participating in the webinar. There'll be a survey coming around, so please take a few moments to share your comments and feedback. This is recorded, so we'll post a link to the presentation recording online in case you want to go back and revisit it. We will try to provide Dr. Anderson with all of your questions. And I'm not going to promise for him that he's going to have the time to address each one individually, but should he find the time, I'm sure you'd learn even more from him via email. So thank you to everybody. Enjoy the rest of your day.
Thank you.